Today to talk to you about my research. We have a nice small session here, so feel free. I'm very informal, so please feel free to unmute yourself if you have a question. You can certainly interrupt me throughout, or if you don't understand something and want me to explain it differently, I'm glad to do so. So my name is Gina Lamandella. I'm entering my 10th year as a professor in the biology department at Juniata College. In addition to being a professor, I'm actually a co-owner of 2 biotechnology businesses that I started here at Juniata with former research students and alum of Juniata College. So I'll be talking to you a little bit about that as well. So the title of my talk is Gut Check, the Role of the Microbiome in Human Health and Infectious Disease. So a little bit about my research story, and I'm sure you guys are experiencing this as well. You know, research and your professional career is not like linear, right? There's going to be a lot of different opportunities that will come upon you, and it's really about, you know, taking advantage of those opportunities and leveraging them for whatever is next. So keeping your opportunities and keeping doors open is really what this is all about. So my research story began when I was an undergrad at Lafayette College, which is a small liberal arts institution in Pennsylvania. And I thought I was headed to medical school. I really didn't know that there was much else you could do with a biology degree. So, you know, I did well in my classes and thought I was headed to med school. And then the summer between my junior and senior year, I was offered a research internship with one of my professors in environmental science. And I immediately fell in love with applied research. And my 2 advisors at the time were like, hey, you know, you can have a career in research and we think you might really enjoy this. So I instead applied to a bunch of graduate schools and was offered a research fellowship at the University of Cincinnati in their environmental engineering and science department, which was exciting because actually, the Environmental Protection Agency was right across the street from University of Cincinnati. So that gave me the opportunity to sink my teeth more into environmental science and applied science projects. But I'll tell you, it was pretty challenging. I was coming from a biology background and I was entering into an environmental engineering program. So I failed my very first test in graduate school and was thinking to myself, man, is this really for me? And so I quickly learned, okay, I just need to catch up on some math and I'll be okay and persevere. So luckily I stuck with it and was able to get a research fellowship at the Environmental Protection Agency. So I was able to do all my coursework at university, and then I was able to work on my applied research projects across the street at the EPA. So there I worked on fecal source tracking. So I designed different molecular markers to detect sources of fecal contamination in groundwater and surface water. You know, fell in love with it, did my master's and PhD in about 4.5 years, and then realized, you know, I had to get good at a special topic like computational biology and bioinformatics. A lot of the data that I was generating was DNA sequence data. And back then, we really didn't have, you know, access to supercomputers and bioinformatics data analysis pipelines. So I said, you know what, I better go build up my skill set in bioinformatics. So I moved out to Berkeley National Lab and the Joint Genome Institute. So those of you that might not be familiar, the Joint Genome Institute is one of the institutes where they sequence the first human genome. So I figured, okay, I'm going to learn from the best of the best. And so I get the opportunity to do a 3-year postdoctoral fellowship out there and learn how to analyze these large data sets. And while I was out there, many of you were probably kids. How many people remember the Deepwater Horizon oil spill, the BP oil spill back in 2010? So when that happened, I was a postdoc at Berkeley. I was working on inflammatory bowel diseases, but then the oil spill occurred. And my supervisor asked if I would go out on a research vessel and go collect some samples right near where the oil rig had blown up. We wanted to study how the microbes were degrading the oil in the deep sea. So again, you know, it's about taking opportunities that present themselves to you. So I joined the research team on the research vessel called the Brooks McCall. I was out there for about 6 weeks' time, collecting water samples, trying to discover what microbes were degrading the oil. And so it was a really neat time because I was immersed with lots of different scientists from different parts of the government. So it was myself, somebody from EPA, somebody from Environment Canada, a few people from NOAA. So it was a really collaborative research environment on this vessel. So I essentially turned a closet in the boat into a lab. Did some lab work there for, like I said, about 6 weeks' time. So it was a really interesting part of my career. So after my postdoc at Berkeley, I then was applying for professorship positions at all different types of schools, research-intensive schools, small schools like Juniata, and I had the opportunity to make a decision. So I was accepted for professorship at a large research-intensive university and Juniata and a couple other schools, and I just kind of fell in love with Juniata because it's kind of the best of the best in terms of you have really passionate students and professors that care intensely about your learning, but also are engaged in really in-depth and impactful research. So it seemed like the best of the best to me in terms of it's a small school, very, very talented students. And so I've loved it ever since. I've been here, like I said, this will be the start of my 10th year. So my background, my technical title in terms of the scientific research that I do is molecular microbial ecologist, which is kind of a mouthful, right? I study microorganisms in their natural environments and how they interact with one another in those environments. And so I fell in love with microbial diversity back in graduate school. And microbes are everywhere, right? They outnumber us, as you'll see in a second. If we try to enumerate how many microbial cells there are on Earth, it's 2.5 times 10 to the 30th. That's a really big number, right? Way bigger than Avogadro's number. So microbes represent an equal amount of carbon, as in all the plants on Earth, 10 times the amount of nitrogen and phosphorus. And we basically are still discovering lots and lots of microbes. We know very little about total microbial diversity on our Earth. About 66%, actually combined, more than 90% of all the microbes out there we know very little about because they exist in the marine or terrestrial subsurfaces. So they're really difficult to access in terms of sampling. And then the other, 5 or 10% of organisms we do have easy access to, and we can profile what microbes are there. The other really interesting thing, and hopefully I'm not grossing you out right after dinner time, but you're more microbial than you are human. If you actually count up all the microbial cells on and in your body, in your gut, on your skin, you're about 39 trillion microbial cells and about 30 trillion human cells. So I began to fall in love with this microbial cloud and trying to understand how these microbes might be involved in maintaining health and how they might be involved in the etiology of many different diseases. So here's that stat for you again. I like to think about this on a cell-to-cell level because we're a super organism, right? These microbes are interacting with our organ systems. They are interacting with our immune system. They are making vitamins. They are digesting our food, making the food bioavailable to our gut cells. They're doing so many wonderful things for us. And when the balance of those microbes gets out of whack or dysbiotic, that's when we see a lot of these disease states pop up in humans. And so I began to start to think about, okay, how can I study these microbial communities? They're really diverse. If you think just about your gut ecosystem, you have hundreds, if not thousands of different species living in your gut. On your skin, it's thousands and thousands of different species. But the classical way that we study microbes is usually by growing them on petri dishes or in media. We try to mimic the environment that they want to grow in, and we try to culture them. And so that's a fine way to do microbiology. However, we can only grow about 1 to 2% of all the microbes out there. We don't know exactly what they like to eat, right? And they love to live in communities like we do. And so it's hard to isolate or cultivate individual microorganisms. So with the advent of molecular methods and DNA sequencing, instead of trying to grow these organisms, we could literally take a sample, any sample, a fecal sample, skin swab, an air sample, water sample, grind up all the material, break up those microbial cells and access their DNA and sequence them. And their DNA is very similar to ours in terms of they have genomes. They're smaller than our genomes, but the sequence of their genomes enables us to use that as a molecular thumbprint, if you will, of who's there. So using their DNA sequence, we can identify and speciate these different organisms and look at the balance, look at who's there, their relative abundance, how that might fluctuate in the context of disease state, et cetera. And so with the advent of massively parallelized sequencing technologies, we're able to sequence millions and millions and millions of these microbes at a time. And the cost of these technologies has come down quite a bit. In fact, pictured here, and we have these sequencers in our lab, we can generate 800 million sequences at a time. And we can actually generate sequence data now real time. This little sequencer is called the Oxford Nanopore MinION. This little sequencer is about the size of a thumb drive, a flash drive, a little bit bigger. Plugs into your MacBook, and you can directly sequence real time on the sequencer. It generates about 1 million sequences within a couple hours time. You know, it sounds like sci-fi, but this is a technology that's here and now. So I always, enjoyed reading some of Hippocrates' work, and one of his quotes always stuck with me when I was a graduate student. It's more important to know what sort of person has a disease than to know what sort of disease a person has. And something that we're discovering, right, is that disease states aren't like these discrete groups, right? It's very fluid in terms of given individuals, how they present with disease, et cetera. So I kind of modified that quote and added microbiome, because now we're trying to understand how this microbiome might put us at risk for different disease states. And can we shift that microbiome back to a healthy or eubiotic state to cure the disease? So that's what I've spent the last decade or so working on. I work on a variety of different disease states through private and federal funding that I've accrued over the years. I've studied the role of the microbiome in things like inflammatory bowel disease, so things like ulcerative colitis and Crohn's disease, diverticulitis, C. diff infections, hypertension, diabetes, heart disease, obesity, necrotizing enterocolitis. So we're really just scratching at the surface in terms of looking at, okay, how does the balance of these organisms, how important is that in maintaining health? And when things get out of balance or dysbiotic, how might that lead to some of these disease states? And so the methods that we use to interrogate these microbial communities are actually pretty simple. In the case of the gut microbiome, we collect a series of fecal samples from diseased and non-diseased cohorts. We grind up that sample and extract and purify the DNA from all the organisms, millions and millions of organisms in that sample. And in this case, we sequence one little gene. It's called the 16S ribosomal RNA gene. And you might remember from your biology classes that all bacteria have ribosomes because they need to make proteins. And this ribosomal RNA gene codes for part of the ribosome. So it's a great target molecule because all organisms have it, but that the sequence of that gene actually is a little bit different for each organism because it's under evolutionary pressure. And so hypervariable regions within this gene will change over evolutionary time. And so by sequencing that gene, we can look at the pattern of their DNA and use that as a molecular thumbprint for who's there. So once we use PCR to amplify that gene, we then sequence it. So we get a list of all the millions of DNA sequences from all these different microbes in the sample. And then a lot of what my lab also works on is bioinformatics and biostatistical analysis, right? We have to take all that sequence data and analyze it, figure out, okay, at this point, it just looks like a series of A's, T's, C's, and G's, right? But who's there? What organisms are there? How many? And so that's what those bioinformatics techniques are looking at. And then we can identify, okay, in disease state, we see an overabundance of, these really bad bacteria, or they're missing these beneficial bacteria. And so we look for biomarkers of these different disease states. Here's one study that I collaborated on with research students in my laboratory here at Juniata, and this actually got published in Nature Microbiology, a top-tier journal. This was a really neat study. What we were looking at is, instead of just a single snapshot of somebody's microbiome, we were looking over time, longitudinally, how people's microbiomes changed with respect to their given disease state. So anybody that's had or knows somebody that has ulcerative colitis or Crohn's disease, for some time, people will be okay. And then all of a sudden, for whatever reason, they will go into this flare-up state. They'll start having, cramping, diarrhea, etc. And so we're really trying to understand, okay, can we predict when somebody is going into that disease state, right? Can we use the microbiome to see, okay, things are getting a little bit out of whack. We need to maybe introduce a probiotic or figure out if there's a way for us to make the microbiome more stable. So what we did in this study was we looked at individuals over 10 time points. Some of them had ulcerative colitis, some had Crohn's disease, and some individuals were healthy. And so I'm going to try and play this video. So what this model is showing is a given person's microbiome over time. And so you can see individuals that have Crohn's disease, ileal Crohn's disease, colonic Crohn's disease, and other rare types of inflammatory bowel disease. Their microbiomes are fluctuating a lot. They're all over the place, right? And individuals that are healthy or have moderate forms of ulcerative colitis, which isn't quite as bad as Crohn's disease, tend to not fluctuate as much. And so we were able to discover what we define as, once the model stops, it'll be a little easier to see. We were able to discover this sort of healthy plane. So when you're over here in this sort of yellow area, your microbiome is in a pretty good state. However, when you go over towards the left, towards this dysbiotic or unhealthy plane, that's when we notice this correlated with people having a flare-up of the disease. So my students went on to build a random forest predictive model to say, okay, are there specific biomarkers that predict when somebody's going to go into that flare-up? And so with over 90% accuracy, they discovered these are the top 6 predictors. These are different bacteria that if you have a higher abundance of these organisms, you tend to head towards that flare-up. So pretty neat in terms of leveraging this technology to help with disease diagnosis and maybe even remission. These are a couple examples of students in my lab. I've mentored over 100 different students over the past 9 or 10 years. And all of them have landed on their feet and are doing something really amazing. And, research is about, developing those building blocks, right? It's not all going to happen at once, right? You're not going to become a success overnight. I mean, I'm still learning at 39 years old. I'm still learning a ton. And so it's about laying those blocks down, laying that good foundation. And so, as you're all headed or even are in your undergrad now, it's about getting as many experiences as you can and layering that foundation. This is Alyssa Grube. She graduated in 2014. In fact, she just came back to Juniata to present her doctoral dissertation. So she's now Dr. Grube. But at Juniata, she did research with me and published her research. She then did a Fulbright fellowship and studied in, I believe, somewhere in South America for a year. She then went to the University of North Carolina and got her Ph.D. there and was funded by the NSF to support her fellowship. She also majored in Spanish as well as biology. And so, she's a great example, as many of my other students, of just layering those bricks and getting as many experiences as you can at each level. It's another one of my students, Nikea Ulrich. Again, some of my most successful students are the ones that are doing a lot of things. She was a double major in biology and German. She received a Goldwater Fellowship, also did a Fulbright and studied in Germany. And then that led her to her PhD that she's working on at University of Montana. She's funded by NASA right now, looking at these rare and really cool organisms, Acariochloris. Again, she spent her time at Juniata. And one of the great things about a small liberal arts school like Juniata is that you can get involved in research really early on. All these students that I showed you started research their freshman year. They didn't wait like me until my junior or senior year to get involved. And so when you have the ability to spend 3 or 4 years as an undergrad, developing those research skills and maybe getting a publication or two out, that makes you, look really good for graduate schools in terms of really competitive and funded programs. And then another example, and I have so many, and I know, we don't have a ton of time, but Truk Lee is another standout student. She graduated in 2018. Truk came to my office as I think a sophomore. She was very shy. In fact, I think she almost passed out when she was asking for research opportunities. So it's another example of push yourself outside your comfort zone. Your professors, especially at Juniata, we have the open door policy. You can come in to our office and chat with us about anything. I keep a big bowl of candy and tissues on my desk for any reason, and that's one of the best things about being a professor is that interaction and mentorship piece. So Truk started in the lab as a sophomore, barely said 2 words. By the end of her research career at Juniata, she had a couple publications and was accepted to the Masters of Public Health program at Yale. Went to Yale, was very successful there and actually came back to Juniata, and she's now a lead researcher with one of my research companies that I'll talk to you about in a moment. And so again, I think in terms of research outcomes here at Juniata, like I said, I've had the honor of mentoring 100+ students. That has resulted in 40 scientific publications with 50 different students. And my students are first author on many of these publications. The other neat thing is you get to travel to conferences, and maybe not during COVID right now, but soon again that will happen. And that's where you really get to discover yourself. I'll never forget my first microbiology conference. You study one little niche, one little area of microbiology or whatever your discipline of interest is, but there's so many other things out there. And so that really opened my eyes to potential opportunities down the road. So I definitely encourage you all, if you can present at a conference, even if it's a local or regional conference, that's great. The more practice you get at articulating your scientific study, the more well-rounded you will be as a scientist. We've also had the opportunity to have over 30 research grants from various governmental and private institutions to support our work. And we are a research family, as you see here. We work hard and play hard, as they say. Another neat thing about Juniata is that we do a lot of course-based undergraduate research experiences or cures. So we don't just do independent research. You have the opportunity to do a more guided research experience in the classroom. And several of my classes are built to do that. And, I have my students take these surveys at the beginning and end of their experience. And these are all the different learning outcomes that were measured. And these are our students in red here. In terms of their ability to garner these learning gains. And you'll see our students in red are well above the national average. And I highlighted a few of the different learning gains where we saw the biggest gains, things like tolerance for obstacles faced in the research process, understanding the research process, the ability to analyze data. Self-confidence, that's a big one. I think research experiences are really good to help you realize how awesome you are. Imposter syndrome is a thing, and it's something you have to grow out of. And I think research helps you build and realize, oh, I can do this. and also learning to work independently was also another learning gain. Okay. I'm going to transition. How much time do I have left? I want to make sure I leave time for questions.
I believe you have till the end of the hour. So I'm guessing another like 15 minutes. So you can section out your time depending on how much you need for questions.
Okay, great. Okay, great. And like I said, if you guys have any questions along the way, just unmute yourself and go ahead and ask them.
Mm-hmm.
So I mentioned in the beginning of the talk, not only am I a professor, I'm a business owner as well. Now, I've never taken a business class in my life. I probably should at some point, but I run two companies. One is called Right Labs. I started that in 2014 with a former student of mine. It was actually his idea. I thought it was going to be a fun little side project that would just kind of peter out. But now 7 years later, we are a microbial genomics company that help different institutions understand microbial communities in any context. That could be, we work with companies on things like biodegradation and establishing biodegradation rates of different microbes. We work with the Environmental Protection Agency. We work with lots of different academic institutions. So at this point, I think we work with over 100 different institutions globally, and research students in my lab, as well as recent grads are actually employees at the company. And then in 2018, we started a new company, myself and Justin Wright, the student, as well as a former Juniata alum, Gary Shope. We started a company called Contamination Source Identification, or CSI. And CSI is an infectious disease diagnostics company. So, I've been talking a little bit about how we study the role of the microbiome in disease etiology. And then I began to think, can we leverage this high throughput sequencing to help us diagnose infectious disease more readily? One of the biggest, hurdles we're going to be up against as researchers and clinicians is diagnosing disease and antibiotic resistance patterns. And so right now, the way we sort of identify disease, it takes days and days and days. And so that's just not good enough, especially in disease states like sepsis, where every hour it takes to get the results back leads to a 5% increase in mortality. So getting really good results, really specific and sensitive results in a timely manner is something I'm passionate about. And so we developed this company called CSI to try to leverage this technology to help with the hurdles in infectious disease. And so we are a startup. We have, I think now 22 full-time and part-time employees. And we occupy about 5,000 square feet of space in our business incubator here down at Juniata College. We also pivoted during the COVID crisis, given our expertise in molecular biology. We got our what's called our CLIA clinical lab license from the Department of Health. And we work with over 15 different nursing homes and medical facilities and schools, helping them with COVID testing. So we do the PCR-based testing. I think we've just reached about 100,000 tests that we've done during the COVID crisis. So it's been a busy time. So like I said, we do the COVID-19 diagnostic testing, but really what the crux of CSI is about is leveraging our patent pending technology that's rooted in DNA and RNA sequencing. And so we're working in a couple different disease states, something that is a big problem in the United States and Europe. I'm not so sure how much of a problem this is in India, but tick-borne pathogen disease detection is a huge hurdle here. Pennsylvania is actually the #1 state for new cases of Lyme's disease each year. And we know with global climate change, these tick-borne pathogens are on the rise and are diversifying. There's over 40 different disease-causing tick-borne pathogens out there to date, and probably some that we haven't even discovered yet. So we're working on developing technology to detect tick-borne pathogens comprehensively. We're also working on another challenging disease state relating to prosthetic joint infections. So hip and knee and shoulder replacements are getting more common, especially as our population is aging. And I forget what the exact statistic is, but I think 25% of people that get a PJI will die within 5 years of their initial infection. And so that's not a great stat, right? 25% mortality rate if you get a prosthetic joint infection. So we're collaborating with the premier Rothman Institute down in Philadelphia. They do, I think worldwide, they do the most joint replacements. So we're working with Dr. Javad Parvizi on developing our technology for detecting PJIs. And so, like I was talking about before, in terms of sort of the way microbiology goes, the current gold standard for infectious disease diagnostics is still culture-based, still trying to grow those organisms. And we know bacteria, we know microbes take, can take a long time to grow. They can take 72 hours up to, weeks to grow, depending on the type of organism. And many times, the actual result will come back culture negative. Nothing will have grown, even though we know there is an underlying infection. And so you can see some statistics here relating to different disease states. So for prosthetic joint infections, the culture negative rate was up to 42%. So 42% of the time, the clinician knew there was an infection, right? But the culture is coming back negative. Same for UTIs, 20% of urinary tract infections come back culture negative. So this is a big challenge. It's a big hurdle in terms of infectious disease diagnostics. And so we're trying to redefine that gold standard by leveraging next generation sequencing to help in terms of timeliness and specificity and sensitivity of these disease diagnostics. So one technology that we are leveraging that seems to be working the best, we have a research paper coming out on this where we do a heads-up comparison of lots of different technologies. The one that seems to be giving us the highest sensitivity and specificity is called metatranscriptomics. So this is essentially RNA sequencing. So instead of looking at DNA, which is great, but DNA, as you guys know, has a really long half-life, right? We can get DNA from dinosaur bones and sequence it. So it's not a good surrogate all the time for active infection. So when we're talking about somebody that has an active infection, we want to know that that organism is alive. And so that's where using RNA as the surrogate is more robust in that if we see RNA around, we know that that organism is actively expressing a given set of genes. And so it's a nice surrogate for activity. And we know RNA has a really short half-life due to the fact that it is single-stranded. So if a cell lyses, usually within about 30 seconds to a minute, the RNA will have degraded. And so this slide just captures that. So our redefining the gold standard, our technology, CSI-Dx, essentially is centered around trying to sequence this microbial RNA from clinical specimens. And so we have a variety of different kits that we send out based on whether it's a blood sample, urine sample, cerebrospinal fluid sample, synovial sample. You can think of really any matrix in the human body. So we have these sample collection kits that have some preservative in them that help stabilize that RNA until it gets back to us in the laboratory. And then in our laboratory, we have programmed these state-of-the-art robots to automate the RNA extraction and preparing that RNA for sequencing. This is really nice because it prevents, like I was saying, we have a microbial cloud that is following us, right? So we don't want to contaminate the samples. And so doing all this robotically in a HEPA filter controlled environment has been really advantageous. And we can multiplex 96 samples at a time on the robot and hopefully acquire more robots over time so that we can do hundreds and hundreds, if not thousands of samples per day. Once that nucleic acid is prepared for sequencing, we load it onto our high throughput sequencers and we generate, like I said, 800 million pieces of data. And so we have, what's really neat is actually a couple of my students have been involved in developing this technology, and we actually have a patent pending to protect all of our proprietary wet lab steps. And so that patent has been accepted. And like I said, I have 3 students that are co-authors on that patent with me. Hopefully this will play. And I think it's got some background music. So this is, I just wanted to show you a little bit about how automation really enables us to parallelize this technology. So this is a Beckman Coulter i7 robot. It's got 2 independently controlled pipetting arms. So the 8-channel pipetting arm will prepare all the reagents for us. And then there's a 96-channel, that 96-channel head will pipette 96 samples at a time. Also on deck is an automated thermocycler to do the heating and cooling steps that we need to prepare the RNA for sequencing. And again, all this is automated inside of the robot. So it took about 3 months to program this, right? I make it look so easy, right? But it took us about 3 months to get all of our wet lab proprietary steps programmed on here. But the hope is that, again, we'll be able to parallelize and speed this process up. So this used to take a lab personnel about 24 hours of hands-on time to do. Robotically, it takes about 4.5 hours. So already we have sort of shrunk the amount of time it takes to process these samples. Because don't forget, clinicians want their answers quickly, right? You want the correct answer and you want that diagnosis as quickly as you can so you can more accurately treat patients. Then the fun really comes. So after all the wet lab processing, again, the other thing that we specialize in is analyzing all of this DNA sequence data to figure out what microbes are there infecting a given sample. And so that's where we have a whole bioinformatics team. It takes all that raw data, filters out the poor quality sequence data, compares it to our proprietary database of more than 10,000 different pathogens. So we have viruses, fungi, and bacteria in our database. What's really neat is that we're also learning from the data. So we do a lot of machine learning and mining of the data to repopulate those databases with emerging pathogens. So many of you know that microbes don't stay stagnant, right? They're constantly changing themselves, like what's happening with COVID right now. It's an RNA virus that's changing quite rapidly. And so the database, I call it this living database because it's growing with how the pathogens are evolving and changing over time. And so we have an entire LIMS system, lab information monitoring system, that wraps around all the wet lab and bioinformatics steps and automatically generates, once the analysis is done, it will populate a report with the specific microbe that is, microbe or microbes that are infecting a sample. And then that gets sent to hospital electronic medical record systems. Again, like I was saying, we're spending a lot of time doing some research and development for identification of tick-borne pathogens. So we're working on our tick-borne pathogen panel. Right now in our database, we have over 110 different tick-borne pathogens. And the nice thing about this technology is that it's comprehensive. It's not like we're looking at one bug at a time. We're able to look simultaneously at hundreds, if not thousands, of different pathogens. And so being able to get that snapshot of the entire profile has proven to be really important. This is just showing some results, how we stack up against more classical molecular techniques. So some of you probably heard of PCR. PCR, I liken this to fishing with molecular hooks. You have specific targets of interest that are pre-designed. And you try to amplify, make many, many copies of those specific organisms. But you can only look for maybe like 5 or 10 organisms at a time. Whereas with CSI-Dx, we're looking at hundreds of different organisms. So this is showing 7 different samples, and these are 20 plus different tick-borne pathogens. And so we did the same samples via PCR as well as with CSI-Dx. And this is a heat map showing you all the different tick-borne pathogens that CSI-Dx found that PCR didn't. So I liken CSI-Dx as fishing with dynamite, right? You throw some dynamite in the water and all the fish come to the top. Same idea here in terms of the comprehensive ability to look for pathogens. Now we're headed towards what's called clinical validation. So when you're a clinical diagnostic lab, you have to do analytical validation, which is, okay, we're going to spike these exact amounts of these organisms in a sample and see how well we do. And then you have to do clinical validation. So you actually have to do this on real clinical samples. So that's where we're headed with our tick-borne pathogen assay. We're getting matched blood and urine samples and comparing ourselves to the current gold standard, which is a two-step ELISA antibody test. And so once we complete our clinical validation, we'll be able to bring this test system to market. Here's a picture of the lab team. We actually our Senator Judy Ward, our state senator, actually came to visit the lab a few weeks ago. She got wind of this technology and is really excited to help us leverage this technology for tick-borne disease detection, because it afflicts thousands of Pennsylvanians each year, as well as in the United States and across the globe. Okay, I want to leave some time for questions. So I think I talked a little bit about all the tick-borne stuff. One last case study I do want to talk about, because this case study kind of changed my life in terms of really realizing how impactful this technology could be. This was a individual that had a PJI, prosthetic joint infection. He came into Dr. Parvizi after, I think, his third revision. So he had, they had pulled his joint apart 3 times, and he comes to Dr. Parvizi with massive inflammation of his knee. Dr. Parvizi says, "Okay, let me take a sample of your synovial fluid. We'll send it for culture. We'll send it for DNA sequencing, and we'll send it to CSI as well." And so in that initial culture and DNA test, the results said, okay, this is a Staphylococcus epidermidis. This is a gram-positive organism, can lead to PJIs. So, if you're a clinician, what do you treat for? You treat for staph epi, right? Well, turns out treatment antibiotic wasn't working. 6 weeks later, he comes back to Dr. Parvizi. He's got so much inflammation, the joint's actually falling apart. Dr. Parvizi takes another sample, does culture, and sends that to CSI as well. And finally, the culture showed, no, it wasn't actually staph epi, it was a multi-drug resistant E. coli. Don't you know, the initial sample that we took from day one, CSI-Dx actually detected that multi-drug resistant E. coli. And so, unfortunately, this gentleman lost his leg due to it. They had to amputate it, so he didn't go septic. And so, it just shows you the power of comprehensive molecular diagnostic techniques like this. And so, hopefully, when we're able to come to market with a clinically valid test, we can help save people's lives. So I want to leave a little bit of time for questions. So please go ahead and interrupt.
I believe there's one in the chat that was sent a couple of minutes ago.
Oh, sure. Let me open the chat up. Sorry.
Mm-hmm.
Okay. Where should I start?
I believe it's the last one. I think we just had one question.
Okay. How should I select my subject?
No, I think
college.
Wait, they might have sent you separate chats. I'm not sure. This one on the general chat.
Oh.
So you can feel free to address them on your end.
Okay. Yeah. So the question I see, is how should I select my subject in college if I want to study about genetics and the human brain? Is that the? Yeah, so that's a great question. I would say if you're really interested in the human brain, neuroscience is probably something you'd want to consider studying. As well as, the great thing about a place like Juniata and other small liberal arts schools is that you can major in multiple things. Actually, at Juniata, you can make your own program of emphasis. So I have students that are like you that want to, mash genetics with neuroscience. And so I'll have them take a smattering of computer science and bioinformatics courses. as well as neuroscience courses and psychology courses. And so, it's all about, finding the right advisor or mentors to help you sort of mash your courses together to support your goals. When is the best time to start research? As soon as possible, as soon as you're ready, right? I think, sometimes I have some students that come from different science backgrounds, and some come to us as very strong science students, and some need a little bit more time to get their wheels going. But if you feel you're ready, I've had many successful students start first or second semester of their freshman year. And at Juniata, actually, your research during the semester counts towards biology credits. So that's really nice in terms of being able to kill two birds with one stone. So yeah, I would say get into it as soon as you're ready, as soon as you can. How do you select a subject if you're interested in research and cancer-related stuff? Oh, that's a great question. So again, I think it's about finding the right mentor. So something I encourage all of my students to do is, meet your professors, get to know your professors as soon as you can. I always say use that time between classes to follow your professor and find out what they're working on, and look up the biographies of the different professors in your department, see who works on cancer research. And, you might be able to do some small projects, at your university, but your professor might also know, I have a vast research network, right? So even though I don't do cancer research, I know about 12 different cancer researchers that do paid internships over the summer at a variety of different hospitals that I can network my students with. So networking is key. It's not just about being smart, right? You're all very smart because you're on at 8 o'clock at night learning about how to do research, right? It's about networking with and finding those right mentors. I think we have one more question here. Is it advisable for an international student to take up a pre-medicine pathway alongside a double major in, I'm sorry, what's ECE and BME? Is that biomedical engineering?
I think so.
Biomedical engineering and ECE would
Perhaps.
be. I would say yes. I mean, so the great thing about medicine in the United States is, we have history majors that go on to medical school, right? We have music majors that go on to medical school. So they're making sure that they take all the courses they need to be able to apply to medical school. But oftentimes, some of our best students are doing double majors in a science and something else to be well-rounded, right? It's not just about knowing the scientific facts. It's about being able to make connections. It's about being able to tell, to discuss your science. And so those other courses outside of science help you with writing, help you with public speaking. So yeah, I definitely think double major is possible so long as you're keeping on track with the courses you need to apply to medical school. Okay, electrical engineering and yeah, absolutely. I don't see why that wouldn't be possible. Again, I think it's about finding the right advisors and mentors for you to help you.
So